Good morning, students. Today we are discussed about recombination. Recombination is topic is considered in genetics for MSc previous students. Recombination of DNA takes place by mutation, exchange of DNA strands, and incorporation of DNA. In this process, the genetic information is rearranged between chromosomes that possess similar sequences. Homologous genetic recombination occurs in eukaryotic at the time of gamete formation during long prophase first of meiosis. Each chromosome has two sister chromatids, each of which contains a duplex DNA. The homologous chromosomes, one is maternal and the other is paternal, pair with each other. Pairing is known as synapsis and involves entire length of homologous chromosomes. Recombinations occurs by crossing over. It involves reciprocal exchange of chromal segments between non-sister chromatids of a homologous pair involving breakage and subsequent reunion in a new arrangement. Chiasma is formed at the site of crossing over. Enzymes like helicases, endonucleases, and ligases are involved. The genetic recombination causes rearrangement of genes, producing altogether new genotypes and phenotypes. These cause variations which lead to evolution. In human, about 30 homologous recombination events occur during each meiosis. The recombination events are much more in bacteria and are even in fungi. The study of meiosis in lily plants by Herbert Stern and Yasuo Hota have provided clinching evidence of recombination. Meiocytes of lily flower buds divide synchronously. It has also been discovered that endonuclease, DNA polymerase, ligase, and other repair enzymes are present in early prophase. Mechanics of molecular level of exchange is studied in detail in bacteria and phages. At present, we will restrict our discussion to the recombination mechanism when DNA strands recognize each other by complementary strands bounded by base pair. In bacteria, genetic recombination occurs during conjugation, transformation, transduction, post-replication, repair, during repair of double-stranded breaks in DNA, integration of fast DNA with chromosomal DNA and transposons, etc. Homologous recombination can lead to gene conversion. Bacterial recombination. Bacteria are haploid, therefore do not undergo meiosis. They possess only one double stranded DNA molecules or chromosomes. There are several types of genetic recombination in microorganisms. The most common recombination is the reciprocal exchange between homologous DNA sequences. During genetic recombination, usually only a part of the genetic material of a donor cell is transferred to a recipient cell. The DNA of the recipient cell and the donor pair with each other and reciprocally exchange DNA strands by crossing over. This gives rise to a new genetic constitution of the recipient cell with new characters. Subsequent daughter cells that contain only recombined chromosomes. There are following main methods by which recombination of genetic material takes place in bacteria. Here is the diagram. In A, pairing of homologous DNA duplex and single-stranded breaks in one strand of each duplex having different alleles, and then holiday junction is formed due to strand inversion, branch migration, elongation of homologous of heteroduplex region, and rotation of holiday junction, and then recombined molecules contain heteroduplex or patch region, no reassortment of flanking genes, and in another molecules are reciprocally recombined reassortment of flanking genes. It is a crossing over product. Homologous recombination. DNA of the donor cell recombines with recipient DNA by reciprocal exchange of DNA strand. The recombining DNA molecules have homologous sequences. The DNA molecules align of pair with each other and undergo crossing over and homologous recombination. The recombinant DNA has new genetic constitution. Bacteria can acquire DNA of other closely related bacterial species and can become transformed. This is known as transformation. Transformation was first demonstrated by Griffith in Diplococcus pneumoniae bacteria to confirm DNA as a genetic material. Homologous recombination is catalyzed by recombination A protein, which is called RAC-A protein. Second is non-homologous recombination. Recombination between DNA segments which have no homologous regions is also quite frequently. Here, the addition or insertion of a small DNA sequences in a recipient DNA takes place. It occurs mainly by jumping genes or transposons. These mobile DNA sequences regularly jump to a new location anywhere on the genome. Often, the transposable elements replicate to generate two copies. One copy remains at the original site while the other jumps to the target site. And third one is site specific recombination. When phase lambda infects E. coli bacteria, DNA of the phage lambda is inserted into the DNA of the bacterium. In this way, phage DNA becomes integrated into the host DNA, or we can say bacterial DNA, and becomes part of the host chromosome. Bacterium containing a complete set of phage DNA is called lysogen. Phage DNA is inserted at a specific site into the host DNA of bacterial cell, and now bacterial cell is called lysogenic cell, or we can say lysogenic bacterial cell. The attachment site of both DNAs possess identical 15 base pair sequences. A phage encoded protein called integrase catalyzes the integration. A host protein called integration host factor IGF is also involved. Integrase is a topoisomerase enzyme which breaks, rotates, and then rejoins the strands of both molecules. In eukaryotes, the site-specific recombination produces antibody diversity in the bacteria, or in eukaryotes it causes antibody diversity in plants and animals. So today in this video we discussed about recombination. Recombination is actually occurred during the process of meiosis at the result of crossing over between the sister chromatids of homologous chromosomes. A chromosome has 4 sister chromatids which are and in homologous chromosome both chromosomes are, one chromosome is come from paternal and one is come from maternal side of chromosome, and during the process of meiosis, at the meiosis first, during the crossing over, the sister chromatids exchange their genes and recombinations occur, and due to the recombination mutation is visible in the different offsprings from their parents, so they are different from their parents. And today we study about 3 types of recombination. First is homologous recombination, second is non-homologous recombination, and third one is site specific recombination, in which phage DNA is inserted in bacterial cell DNA at a particular site. That's why it is called site specific DNA and it occurs only in bacterial cell and in some eukaryotic cells also. Thank you, students.